On the surveillance and diagnosis of vector-borne viral infection. And I have already seen that among the audience there are a lot of friends, experts in the field, so maybe some things that I am presenting are well known. Anyway, I want to introduce the topic and then provide some updates on the epidemiology and which are the challenges in the diagnosis and how the PREPARETiD project is contributing to the improvement of rapid diagnostics. I am also representing the European Reference Laboratory for Public Health on Vector-Borne Viruses. I have seen that there are colleagues from France, so Remy Charel, and the Netherlands, Lance Presser, maybe any other that is who are joining these webinars. So we are also providing a lot of support also as European Reference Laboratory to improve the diagnostics of this infection. Regarding vector-borne disease, these are very relevant and are represented a global threat. Over 6 billion people worldwide at risk to develop a disease in their lifetime. Over 300 million cases of vector-borne disease are reported annually, including not only viruses, but also parasites, bacteria, and are associated with a high mortality rate. Vector-borne viruses mainly circulate among wild animals, which represent the amplifying host, and they are transmitted among animals by vectors, mainly ticks, mosquito, midges, and phlebotomus, while humans and other mammals represent the dead host and are incidentally infected when they enter into contact with this enzootic epizootic transmission cycle. In some cases, these viruses are fully adapted to the human host, which represent the amplifying host. They develop a high-level viremia, so they can transmit the virus to the mosquito. Mainly Aedes mosquito species are involved in this transmission cycle. The virus, the most common viruses are dengue, yellow fever, chikungunya, and Zika in the so-called urban epidemic transmission cycle. This is a figure from a quite dated paper, which was published in Lancet in 2012, which showed the rise number of cases reported for vector-borne disease, and the situation is even worse now with the effect of climate change. This slide and also the next represent the geographical distribution of the most clinically relevant vector-borne pathogens, viruses, which belongs to three main groups, orthoflavivirus, alphavirus, and the different genus within the Bunnevirales order. Among the orthoflaviviruses, for sure, dengue, yellow fever, Zika are associated with a high number of outbreaks and human cases of infection, and are mainly distributed all over the tropical areas, while other viruses, so West Nile virus, tick-borne encephalitis, are distributed in more temperate region and including also Europe. Among the alpha viruses, the most widely distributed and associated with a higher number of cases is chikungunya in all the tropical areas, but other relevant, possibly the next emerging viruses are Onion and Mayaro virus, respectively from South Africa and the Americas. The equine encephalitis virus are distributed in the Americas and have been recently associated with the outbreaks in South America, and especially the Western equine encephalitis virus. Sindbis circulates in the northern part of Europe and South Africa and is associated with the febrile illness, but its presence also in European countries is probably underestimated. Among the Bunyavirales order, Toscana virus circulates in the Mediterranean area, as well as other flaviviruses, and is associated with dry illness and meningoencephalitis during the summer period. The Crimean-Congo Hemorrhagic Fever virus will be addressed later in the next talk, and another important virus associated with the high mortality rate is Rift Valley fever. Ropush was quite a neglected virus in South America that has recently expanded its geographical range associated with the new previously unnoticed clinical presentation, and with some cases also imported in Europe. But there are also many other orthobunia viruses which circulate in Europe and could be associated with human disease. So the scenarios of the vector-borne viruses is quite complex, and any of these viruses could be relevant and be the cause, the etiology of the nice large outbreak, even at large scale worldwide. So in this situation, having the availability of a platform that in an unbiased way can detect these pathogens really relevant, and is the core of the PREPARETiD project, a EU-funded project that has the aim to develop rapid diagnostics for in the emergency setting, like the next disease X pathogen, within a One Health approach. So it is based mainly in a mobile lab with a metagenomics sequencing approach based on nanopore sequencing, which can work also offline through a dedicated pipeline for data analysis. So it has already been tested in different settings, and it allows to detect the pathogens. Then once the sequence of the pathogen is identified, then other tools can be applied. So specific target-specific test that can be used as a point-of-care test, or the target can be introduced in a multiplex, the so-called One Health pod that can be applied to different types of samples, environmental, animal, vector, or human samples, with a multiplex approach or high throughput approach. But at the core of all these diagnostic techniques, there is a novel extraction technology that is very fast. It can be adapted to different type of material, and it provides a fast and highly sensitive approach for the isolation of nucleic acids, and also starting for a very tiny amount of samples. And then there is integration in the digital One Health platform. So these topics will be further discussed by Andy in the next presentation. So I'm moving on, describing what could actually be the scenario or what has already happened in the European countries. So vectors that are competent for the transmission of these exotic viruses are already present in Europe, and they are spreading, they are expanding their geographical range, especially Aedes albopictus, and there is the risk of introduction of Aedes aegypti that is even more efficient in the transmission of these viruses. And during the last almost 20 years, several outbreaks of chikungunya, dengue, and even Zika have occurred in Europe, and these are the number of cases that have been reported during the different years in the European countries, with also huge outbreak in Italy in the past, but also in the recent years, and a meta-analysis of all the local transmission events that have occurred in Europe for dengue, Zika, chikungunya show that there is an exponential rise in the number of local events of transmission, but also the total number of cases. And the problem also that was observed in this meta-analysis was a delay in the identification of the primary case of the infection and also the index case of infection, of course. And this can lead to a delay in the implementation of control measures. In Italy, our lab identified for the first time a local transmission of dengue. It occurred in a rural area. Unfortunately, at the time, only a few cases were infected and occurred in the family house household, so in a family setting, and some other cases in a neighborhood. And the rapid identification of the case and the implementation of vector control measure allowed to reduce probably the size of this outbreak. And this is the timeline of this outbreak with the identification of the index case, who was a family member of the primary case returning from Indonesia. So in this case, it was possible to identify the primary case. And this slide shows also the timeline of the detection of the viral RNA in blood. And urine was also a useful sample, which allowed to diagnose cases also in late phases of infection after a long time since the onset of symptoms. And the length of the outbreak was quite long, a couple of months, and this is in line with the climate condition and in Italy, even in the northern part of Italy, where the time when the vectors are active and can transmit the virus is quite long. And this slide shows also the intervention, so the mosquito control measure, but also the entomological survey with testing of the mosquito, collection of the mosquito, the epidemiological investigation with the 8 active case search, the rapid, the immediate implementation of screening by nucleic acids method of blood, cell and organ donors at the province level, and then again entomological survey to check if the control measure were effective. Full genome sequencing of the virus was also helpful to link this outbreak to the primary case with introduction from the Indonesia, since the full genome sequence of this virus cluster together with other strains circulating in the area. So this timelines for epidemic course shows the different number of cases, many imported cases recorded in Italy in the last 10 years, and this is the autochthonous cases reported in the outbreak that I mentioned. This outbreak occurred in 2020, when at the time of the COVID pandemics, with travel restriction and also restriction in the movement of people, so it was quite unusual and unexpected that a travel could come from other endemic country and import the virus and be the starting point for a local transmission. Anyway, it occurred, but in the following years, this risk, of course, increased with increase in the number of travels, but also because of the international situation with a significant, dramatic rise in the number of cases, especially in South America. This is for dengue. And this was associated with a higher number of imported cases. And this was associated also with a quite large local transmission event in Italy. So this is the epidemic curve of a transmission event in the Marche region. This was really an outbreak with about 200 cases, including the confirmed and the probable cases. And as you can see in this slide, the identification of the index case was quite late, about a month later than the probable initiation of the outbreak. So it is very important to early identify this outbreak, these imported cases and the first cases of autochthonous transmission to avoid this large outbreak. And this is the general data from Italy regarding dengue in the last year. So there was a quite large outbreak in the Marche region, but also several other cases in different regions in Italy that are highlighted here. And so over 200 cases were confirmed, and 480 cases, so over 200 cases were autochthonous and confirmed, and 480 cases were imported. Similar situation for chikungunya now in Europe. So there are several local transmission events reported in France, with a total of 30 cases reported in the ECDC website till July 16. One case is also from Italy, and this corresponds to an international situation with a large outbreak that has occurred in La Réunion, 18 years after the previous large outbreak that occurred in the Indian Ocean, and this, of course, is associated with a higher risk of importation of the virus through viremic travelers. And the first actually autochthonous transmission of exotic virus occurred during the outbreak in Indian Ocean and La Réunion in 2007 from a traveler coming from India to Italy in this region in the northern Italy, in Castiglione di Romagna and Castiglione di Cervia, two small villages, but a single introduction led to a very explosive outbreak with over 300 of human cases of infection. And even in this case, there was a delay in identification of the first case and implementation of vector control measure. And this was so that the outbreak was identified mid-August, while the index wolf case occurred in June. After 10 years, again, other huge outbreak of chikungunya explosive occurred in Italy. This first outbreak occurred in Anzio, in Lazio region, and the first case was identified in the beginning of September, but probably the outbreak started in June. And this led also to a secondary outbreak in Rome and also in the southern part of Italy, in Guardavalle Marina. So the total number of cases was about 500. There was also one death confirmed due to the disease. Several cases were also hospitalized. So this is something that can happen anytime in a European country, and this is an effect also of the climate change. Why there is this delay in identification of this outbreak? The problem is related to the symptoms of this arboviral infection that are quite non-specific. They overlap with other arboviral diseases and also with other other infectious disease. And this slide summarizes indicates the different viruses that can cause, among arboviruses, cause neurological syndrome, hemorrhagic syndrome, arthralgia, rash like chikungunya, or we should also remember that most of these infections are asymptomatic and are, of course, unrecognized because people do not report the symptoms. And then there is the problem of the overlap of symptoms with the other viruses that are endemic in Europe. This is the case of West Nile virus, which is causing outbreaks in practically all the European countries, of almost all European countries. Most cases, human cases, are reported in the southern countries, so Italy, Greece, but also Romania, Hungary, Serbia also are reporting a lot of cases. Wetland area, bird flyways, so migratory route and height of birds and high temperature are all risk factors for the expansion of the main vector, the Culex mosquito, and hence also the virus. A virus that circulates among wild birds and can transmit incidentally by the mosquito bites to humans. It is associated with neuroinvasive disease in the most severe cases, most commonly with fever, but most infections are asymptomatic, about 80% of the cases. These are the cumulative number of cases reported in the European country to the ECDC. That shows a huge number of cases, a huge outbreak all over Europe in 2018, but there is also a rise in the number of cases in the recent years, showing a trend towards increasing the number of outbreaks of cases and also geographical expansion of the virus. In Italy, the surveillance plan for West Nile virus is integrated with Usutu virus infection surveillance. Usutu virus infection in humans is also notifiable, even if it does not represent a trigger for the implementation of screening of the blood donors to prevent the transmission through donation of substances of human origin. But the reason for this surveillance is because we want to understand more about the impact of the Usutu virus infection on human health. However, based on the data that we collected during time, these are from the Veneto region where I work, the number of human cases is very low, so this confirms that the virus is not very pathogenic to humans, while the number of human cases of West Nile virus infection is much higher. We reported over 1,200 cases over the last 15 years, and there have been huge outbreaks in 2018 and 2022. And this corresponded also to an increase in the number of detection in the mosquito. through the surveillance. Regarding the One Health approach to diagnosis, this is very relevant. This is the basis of the Italian surveillance system and in several other countries in Europe. And it is based on the surveillance in high-risk areas of the positivity, so the presence of the virus by molecular techniques in mosquito pools, in wild birds, also the presence of disease in equids, and of course the human cases. These maps and also epic curves shows two consecutive seasons, 2022, when there was a huge outbreak in the northern part of Italy, and the following years in 2023, when there were a lower number of cases, and there is a correspondence between among the three surveillance systems. This is useful, this surveillance, because it allows to implement control measures, so vector control, information to the population, and especially the implementation of nucleic acid testing for West Nile in affected areas, when only where there is evidence of circulation of the virus and when there is evidence of circulation of the virus. So this allows also to reduce the cost of testing and allocate resources when and where they are needed. The large outbreak that occurred in 2022 was associated with the record temperature, so very high temperature and very low precipitation that allowed the expansion of the virus between birds and their vectors, but was also associated with the introduction of new strain of the virus, and this was identified through full genome sequencing of the virus. This allowed to track the introduction and the spread of the virus in the region, and also to have some important information about the correlation between the genotype of the virus and the clinical presentation of the disease, because we observed a very an outbreak with a lot of severe disease of patients with a severe clinical presentation, especially with acute practice paralysis, and this was associated with this lineage 1 strain. Regarding the laboratory diagnosis, I'm showing you this table for the publication that describes the local outbreak of dengue that occurred in 2020, where I would like to highlight that these are the CT value of the PCR, real-time PCR that was done in different samples, so blood, urine, and saliva. Sometimes the viral load is quite high, such in this case, but in most of the cases, the viral load was quite low. And so we need a sensitive method. Regarding the serology, you can see here that these are the OD value of the ELISA assay, that there is a lot of cross-reactivity in the serology test, especially for the IgM, but also IgG antibody between different flaviviruses, in particular here between test ELISA assay for dengue and West Nile virus. And this represents a problem because if we based our diagnosis only on serology, these dengue cases could be misdiagnosed as West Nile virus cases. These are all dengue cases without a travel history. So it is very important to use molecular testing, to have a very sensitive test for the identification and confirmation of these cases. And because of the broad cross-reactivity of serology tests, we always need a confirmation by a neutralization test that can be done in experienced laboratory, in reference laboratory, and need the parallel testing of the different viruses which are considered in the differential diagnosis, and it's required times also, so with a delay in the diagnosis. For dengue, NS1 testing is very useful because it can rapidly identify the cases, but we should also keep in mind that that has a lower sensitivity than the molecular test, as shown also in our experience with several cases who were positive by PCR but negative with the NS1 testing. We should also keep in mind that the molecular test is positive also in cases who have been recently vaccinated. The viral RNA is detectable in blood and urine, and we should remember also this this point, because the vaccination is based with a live attenuated virus. Regarding the laboratory diagnosis of West Nile virus infections, it's even more difficult than dengue virus infection, and because of the low level viremia, so we need a really very sensitive method to detect the virus in the blood. Testing viral RNA in urine can be useful because it can expand the time window when we can have a positive test. This slide summarizes the data that we collected during several years about the results of PCR testing in whole blood, in urine, in different time points since the onset of symptoms. And in our experience, about 70% of patients with a neuroinvasive disease or with fever have a positive molecular test in their blood, in their urine, in the first week after symptom onset, and the rate of positivity decreased during time. Anyway, there is a large percentage of individuals who are still positive even after 2 weeks after the onset of symptoms. Regarding serology, if the patient is immune compromised, of course, the patient do not develop an antibody response, so the serology is not very useful. Generally, in immune competent individuals, they at the time of symptom onset or after 1 week after the symptom onset, there is the development of IgM and IgG antibodies, but we need all this confirmation through neutralization assay. And then we should also keep in mind that if these individuals have a prior immunity against a related virus belonging to the same genus, there could be an anamnestic response, so-called original antigenic scene. So in this case, they only develop IgG antibodies, even at very high titer, but they do not show a rise of the IgM antibodies. When we faced the large outbreak in 2022, we had also to reorganize the work in our lab, because we had to face of a huge workload with a lot of samples arriving from all the region. And and it was also the period of July to August when also the technician and the personnel is in vacation. Anyway, we were able to use high throughput platform like the one that is used for the screening of the blood donors, with a very highly sensitive method for the detection of the viral RNA. And this allowed also to reduce the turnaround time to get results, and this is very important to inform the public health. And so these are our experiences. So with a very low turnaround time during the peak of activity in August, and in over 40% of the case of the test of the results had a turnaround time of less than 24 hours. So in conclusion, in Europe, the number of detected local outbreaks of Aedes-borne viruses has progressively increased in recent years. Moreover, there is an expansion of the endemic viruses in Europe, both geographically and as in number. So we need to improve our diagnostics, because the diagnosis is very challenging because of this immunological background of the population, that challenges especially the serology testing, but also we need a very sensitive method, because also the molecular testing is not easy because of the low viral load. And this it is important to recognize very early this both the imported cases and the autochthonous cases in order to prompt activate public health intervention. And the rapid One Health diagnostics tools that are being developed, we are working a lot also as a European reference laboratory, but also with the research project like the PREPARETiD are very important to improve the diagnosis.
Good. So then, thanks a lot for for attending. So in so many numbers, shows us that this webinar series around the PREPARETiD project seems to hit quite some interest. So after this extremely insightful talk of Luisa, I'm looking more into some technical aspects. So it's very technical oriented and is particularly focusing on a rapid extraction method that we had developed here at Xpedite Diagnostics, and many aspects of that development also came through the PREPARETiD project, which is an EU-funded project where Xpedite is just one of the one member of the consortium. And the main theme of this EU project is to develop new tools for essentially pandemic preparedness. And we can roughly intersect this in three different pillars. One is a molecular home testing on infectious diseases, mostly viral diseases. Another one is an from sample to result, so an integrated multiplex qPCR-based system. And also another very important building block is a mobile suitcase lab, which enables you to do nanopore next-gen sequencing in a mobile fashion, so outside of centralized labs. And as you can imagine, if you receive a sample, that needs to be processed in a certain way to make the contained nucleic acids accessible to those molecular tests. And that was the task here at Xpedite Diagnostics. So we have developed and are still developing different rapid RNA and DNA extraction methods and protocols that feed into these different molecular analysis methods. So before I go into some more detail on the data that we have gathered so far, I'm just doing a very brief wrap up of what's so special about the way that our extraction products are working. So we call it the SwiftX way, and what is different? There are several aspects that are different. First of all is that we use magnetic bead technology to capture cells from liquid samples. That can be complex liquid samples like sputum or whole blood or urine. So whenever we have those liquid samples, the first step is not to just lyse everything that's inside and trying to get hold of the nucleic acids, but instead we keep everything alive and intact and bind those biological cells with our magnetic particles, pull them apart, remove the matrix, and by that we have a concentration effect in that case. And these captured cells are then in a second step lysed, destroyed, disintegrated by different methods. So we have also a portfolio of lysis methods, lysis buffers to do this. We can tailor this to the cell type usually that we are facing and we want to extract from. And the last step is sort of purification, but it's not the standard way of purification where you would bind the DNA or RNA to a solid support and you would wash and you would elute. No, what we do is what we call reverse purification. These magnetic beads that we are employing here are not binding DNA or RNA. They are binding debris and proteins and other inhibitory substances that could interfere with the downstream molecular analysis. So that's why just a one-step procedure, very fast but still powerful. So to wrap this up, first, different method is capture of cells that works for all bacterial and fungal cells, also parasites. Second step is an lysis approach that is not based on caltropic salts. And the last one is this reverse purification. It's a cleaning of the lysate, of the raw lysate, which, because we never bind nucleic acids, allows us to do a purification without loss of RNA and DNA, which is of particular interest if you have some samples with extremely low cell counts, for example. And this method, which I'm going to present in the next few slides, which we have called SwiftX virus, is only using the second two pillars of that workflow. It's a one-step DNA RNA extraction method. And here in the next slide, I show you a little bit more how it works. So if you, so the method is intended to be used for blood plasma and blood serum. And the chemistry behind the kit is very simple. It's these magnetic beads that I've mentioned for the reverse purification. It's a lysis buffer, which is not a chartropic buffer, but it's compatible with molecular analysis, and it is proteinase K. So the protocol foresees that you mix these three components with your sample, which is, as I said, blood plasma or serum, and incubate at high temperature, in this case 95 degrees. And what happens during that is, first of all, we inactivate nucleases, RNases, DNases, to render the released nucleic acids safe. And of course, likewise, we also disrupt viral particles, but also host cells with that conditions. And after that, this vessel is transferred to a magnetic separation rack, or if you can also, of course, automate that procedure, but it's very simple. It's a one-step process, so automation is not really needed still. So after a magnetic separation, what happens here is what I mentioned. We remove inhibitors, mostly high molecular weight compounds from this raw lysate, and what remains in the liquid and the supernatant is the DNA and RNA, which is then really directly accessible to molecular analysis. So the whole process takes just 6 minutes. It's very simple, yet powerful. And since it is so simple, and I showed you it's not including this first cell capturing step, there are some limitations to the process. So one is we use a rather small volume of a sample. We do not have a virus enrichment step included. And that's why the analytical sensitivity is a little lower than if you look at let's say, a QIAGEN column extraction, where you have a certain volume of plasma that you put in and a much lower volume that you use for the elution. Nevertheless, there are a couple of advantages. First of all, since it is very simple, it's really well field applicable, especially in this mobile soot gas lab, which I showed you a few slides ago. It's almost instrument free. You only need a heating block and a magnetic rack. So there's nothing complicated that you need to carry with you. It has a secure inactivation of nucleases included. Amplification inhibitors are removed by this reverse purification step. Yeah, and as I said, the whole process is really, really short. So after finishing the development, we had started to gather validation data of the kit. And for that, we performed studies at 4 external sites using leftover samples. And during that, or within these studies, we benchmarked the extraction method against various other established reference methods, like a QIAamp viral RNA kit, a MagnaPure kit. There was an OptiPure kit. There was an Indical pathogen kit. And also different PCR assays have been used for the detection, which also proves that indeed the lysate that is yielded by the method is compatible with different amplification chemistries. So we did some analytical testings with spiked blood plasma and sera. Those were about dengue virus spiked samples, chikungunya spiked samples, yellow fever, and also Lesotho virus spiked samples. And along, we also tested a number of clinical samples. Was a total of a little bit more than 100 samples, inherently positive for dengue virus, for West Nile virus, Crimean-Congo Hemorrhagic Fever virus, and also Leptospira. And in the next slides, I'm showing you a few details on those tests. But before I do that, I need to introduce a little explanation, because what I told you, With our method, we have a sort of dilution factor, which leads to a lower analytical sensitivity. And if you, which you cannot avoid because it's part of the method, as it is part of the method of a conventional extraction, that you do have a sort of concentration effect. So to really evaluate from an analytical and scientific point of view how well the viral nucleic acids are retrieved and how good the yield of the method is, we need to normalize the CT values that we get out afterwards. And that is basically done by simple calculations. So here I have an example. You can imagine a plasma sample with a titer of about 10,000 viruses per milliliter. And if you then look into different methods, let me use the laser pointer. Different methods which use different sample inputs. For example, the SwiftX virus method uses 20 microliter of plasma as an input, whereas, for example, a Kaya-AMP method uses 140 microliter of sample input, and others. So that simply means if you have this titer of 10,000 viruses and this amount of sample input, then you would try to or attempt to extract, in the case of QIAGEN, 1,400 viral particles. In the case of SwiftX, about 200 virus particles, because that is what is contained in this amount of sample. Now, if we go through the process, for QIAGEN and also here the Roche MagnaPure as another example, you would elute your purified nucleic acids in a certain elution volume, which is, let's stick to QIAGEN here, is an elution volume of 60 microliter. Whereas for the rapid one-step extraction method by Xpedite, you had mixed this 20 microliter of sample with the buffer, the lysis buffer, with this magnetic beads and with the proteinase, so that at the end you have a total volume of 138 microliter. So if you assume 100% extraction efficiency, then you would still have at the end basically the same number of viral particles that you had in your sample in this elution or lysate. However, since we do have also different volumes here for elution or total lysis volume, these numbers would yield quite a different virus concentration in the end. So whereas if you think about extracting 1,400 particles with Kaya-AMP, eluting it at 60, that would simply calculate to 23 viruses per microliter of this eluate. Whereas with this rapid method, you would have these 200 particles in a total of 138 microliter, which is then 1.5 virus particles per microliter of this eluate. And you can see by that, if you subject the same amount, let's say 10 microliter of these lysates or eluates into a PCR, then you automatically have a certain CT difference, which is inherent to the methods, between the different methods. So in the case of the QIAGEN, which is, as you see, a 16-fold difference in concentration that transfers into a delta CT that you have to expect of 4.01. So now, if we look at the data with spiked samples, what you see here is the CTs normalized. So the CT of the here upper row is going to be the Kaya-AMP viral RNA extraction compared to the SwiftX virus extraction, and the lower row is the MagnaPure method versus SwiftX. So, and this is basically spiked plasma samples spiked with chikungunya, in increasing dilutions. You see that the CT value is also increasing with the dilution. That's, of course, totally expectable. And what for better comparison of the methods, we have now adjusted the SwiftX method by this 4 CTs for the Kaya-AMP and 2-point-something CTs for the MagnaPure. So means whenever you see here both methods showing the same CT value, that means that the yield of the RNA extraction is the same, because it's dependent on the input volume and the output elution analysis volume. So for chikungunya, you see indeed a very nice match of SwiftX virus and the QIAamp RNA kit. Whereas for the MagnaPure, you see that the SwiftX virus gives slightly better normalized CT values, indicating that the preservation of RNA or the retrieval of RNA is a little bit better. However, as you see in the next diagrams here, that always varies a little bit with different viruses. This is another example of yellow fever virus spiked into plasma. There is indeed quite some distance or difference between the QIAGEN CTs and the SwiftX CTs, indicating that yellow fever virus is not extracted with, let's say, 100% efficiency. However, with the MagnaPure, you see it the other way around again, that the CT values, the normalized CT values, indicate very good retrieval compared to the MagnaPure for yellow fever virus. For Osutu, again, it's rather close if you look at the QIAGEN, and interestingly, again, rather big gap between the direct extraction and the automated Roche solution. So that were spiked samples, and the next slide will show you data from two different studies or tests that we did with clinical specimens together with our collaborators. So first study was on the number of dengue virus positive samples. It's a total of 11 samples here from Luisa's lab in Padova. So you see here, and this is this time non-normalized CTs, so it's purely extraction with the respective method, subjection of eluate into the PCR, and the CT you see here on the x-axis MagnaPure CTs, on the y-axis SwiftX virus CTs is what you get out. So you see there is clearly a correlation. However, you also see that there is an offset, yeah, from the yeah, how would you call it, from the middle, basically an offset, because there is an expected delta CT towards the direct extraction method due to sample dilution. Nevertheless, all samples being tested positive with the MagnaPure were also tested positive with the rapid extraction method. And this diagram here is showing a similar study that had been performed in Professor Nazif's lab, who is unfortunately not able to join today in Sivas in Turkey. This is about Crimean-Congo Hemorrhagic Fever virus positive samples. So we tested a total of 50 positive plasma samples. And you see also here, this is referenced against the in-house extraction method and amplification method of the local hospital versus the Swift extraction, the SwiftX virus method. Again, you see some sort of correlation. However, again, there is the expected offset from this middle line here. But interestingly, you also see that this offset is not the same throughout the range of CT values that we get with the in-house extraction. Now, you see that this is sort of getting closer and closer towards the actual on par extraction, on par CT value, the higher the CT value gets. And we had to look into that, try to find a reason why this is, why the CT shift is so dynamic. And we looked into very different features. Before I come to that, just concluding on that, based on these studies, we see still 100% clinical sensitivity for this dengue virus positive samples, and the same for the CCHV positive samples. And likewise, we also tested a number of negative plasma samples, which also were 100% matching concordant with the reference extraction, indicating that Despite it's a very simple one-step extraction, it does not introduce any unspecificity to the downstream PCR methods. So coming back to this weird changing delta CT behavior. So this histogram here is showing you the delta CT that we had observed for the CCHV samples. This is the expected range, based on the input, output, and subjection to PCR. With this particular method, the expected delta CT was around 7.5, somewhere here. But what we actually observed was quite a range, and interestingly, quite a bit of samples having a lower delta CT than you would expect. So the question was, why is that? So we looked at many different things, but to cut the story short, we found the reason or a nice behavior according to or dependent on the delta CT value of the initial characterization by the hospital's extraction and PCR. So again, this is CT values of the reference method, and this is the delta CT between reference and SwiftX virus. You would expect, if everything is just according to math, a delta CT of 7.5, something like in this window here. But what you actually see is with low CT values, delta CTs that are larger than expected, and with higher CTs, delta CTs that are lower than expected, which leads us to the conclusion that with samples that have a high viremia, so low CTs, the swift extraction performs not as expected, so definitely worse. But whereas in samples where you have just low viremia and not so many virus particles, it performs actually better than you would expect, or maybe to put it vice versa, the reference extraction is not working as well as you would intend. So that was an interesting behavior. And with that, I'm actually almost at the last slide here. This is just an overview again. So this SwiftX virus kit with a very simple setup is a one-step protocol for RNA DNA extraction, which is also very short with 6 minutes total extraction time. The advantage is you still have a purification step in it with this magnetic beads here. So we really actively remove blood plasma inhibitors. We had now validated it with more than 100 clinical samples throughout different virus species, including analytical testing with spiked plasma samples. And we do plan further that we want to optimize it also for the nanopore next-gen sequencing, especially important for the PREPARETiD project, where we need to couple this field applicable rapid extraction with the nanopore sequencing to enable virus strain sequencing in the field. And what we also plan is to release a CE-IVD registered version by hopefully next month, by August, to also make it accessible to routine diagnostic testing. So last but not least, I want to send a very big thank you to our collaborators here from the PREPARETiD project. That is foremost Dr. Ahmed Abdelwahed from University of Leipzig in Germany. That is Dr. Sarah Savic and Dr. Tamas Petrovic from the Veterinary Institute in Novi Sad in Serbia. It is Professor Nazif Elaldi from Sivas in Turkey. And last but not least, it's Professor Luisa Barzon from the University of Padova in Italy. So thank you very much for giving us the opportunity to really test the product and optimize the product with you.
It is a land where all three continental cultures intertwine in Turkey. and its disadvantage is that it serves as a pathway for the transmission of diseases, especially zoonotic ones, between these three continents. Our main topics today, Crimean-Congo hemorrhagic fever is a viral disease characterized by fever and hemorrhage. While the first serology, can you see also again?
Yeah.
Oh, thank you very much. Crimean-Congo hemorrhagic fever virus, a member of the Nairoviridae family, and virus also. And genome is composed of three single standard negative sense RNA segments, known as small, medium, and large. CCHF is listed as a priority pathogen by the WHO, with no approved vaccines, therapeutics, and point-of-care tests. The virus is transmitted mainly by the Hyoloma ticks species, exposure or throat contact with blood or tissue of infected animals and humans. As you know, this is defined firstly in 1944 in West Crim. It was shown in 57 countries for now in three continents. Turkey is most affected country, and a new outbreak is defined in Iraq, and Crimean-Congo hemorrhagic fevers focus on now Spain, Portugal, treat EU. While the first serological diagnosis in Turkey was made in 1982 in Aydın province, Aydın city province, and located this city located with west most part of Turkey. The first virological diagnosis was made in the spring and summer of 2002 in the Tokat province, located in the central Black Sea and central Anatolia region of Turkey. After the first case in 2002, it has become visible in almost all geographical regions of Turkey today. You can see in this map also, and concentrated in the valley we call Kelkit Valley, which passes through the borders of a lot of cities like Amasya, Yozgat, Tokat, Sivas, Gümüşhane, Giresun, Erzincan, and Erzurum. And you can see Kelkit Valley and in the north of central Anatolia and in the south of Black Sea region also. You can see this also separated to all Turkey, but mainly cases in this area, Kelkit Valley. Yeah, this graph shows the number of cases and number of dates over the 22-year period between 2002 and 2024. As can be seen from the graph, there was a significant increase in the number of cases after the first diagnosis in 2002 and until 2008 and 2009. However, as the severity of this disease was recognized and necessary precautions were taken during this period, and the number of cases decreased due to the COVID-19 pandemic time in 2019, 2020, and 2021. This number of cases yearly doubled, you can see. However, due to the climate conditions and the measures taken, the number of cases has halved also in the last 2 years. This demonstrates that the precautions and measures can be taken against these diseases are effective. And we can see that the mortality rates in the countries with the highest number of cases worldwide, although Turkey has been among the countries with the highest number of cases in recent years, we see that it has the lowest mortality rate. You can see and it showed that 5.6 also. This demonstrates that knowledge base surrounding Crimean-Congo Hemorrhagic Fever in our country has developed in the clinical and intensive care treatment and approaches. And Sivas Cumhuriyet University Hospital and have shown it also is a very big hospital and 1,200 bed research hospital with all inpatient clinics and outpatient clinics. Our laboratories, both in microbiology and all other fields, are world class. Our hospital is one of the reference hospital for Crimean-Congo Hemorrhagic Fever. Our hospital serves approximately 1,200,000 people and is one of the largest hospitals in our region. Until 2024, 2,695 CCHF cases were followed at our hospital. Patients in our region, especially those with moderate and severe conditions, are referred to our hospital and treated in inpatients. In addition to our hospital's laboratories, our university also has a large scientific research center. Scientific studies on Crimean-Congo Hemorrhagic Fever are conducted in this laboratory by a team led by Professor Dr. Nazif Elalda. And Cumhuriyet University and Sivas Province, where we work, as located right in the middle of this valley, and we are the province where Crimean-Congo Hemorrhagic fever is treated the most also. And we can see this graph shows the number of cases and rate over the 22-year period between 2002 and 2024. As can we seen from the graph, there was a significant increase in the number of cases after the first diagnosis in 2002 and until 2008 also. However, with the recognition of the seriousness of the disease, the necessary public information and implementation of preventive measures during this period, the number of cases remained lower than also. Yes, and okay. The slides show the number of cases treated at our hospital from 2003 to 2024. Case numbers have increased or decreased in parallel with the number of cases in our country and region. Over 22 years, almost 2,695 cases of Crimean-Congo hemorrhagic fever also treated in our hospital also. Yes. And the slides analysis of 205 CCHF patients in Cumhuriyet University Hospital in 2022. Distribution of cases by months also. And in this is characterized of 2005 Crimean-Congo hemorrhagic fever patient treated as inpatient at our hospital in 2022. Of the 1,310 cases have seen in Turkey in 2022, 205 were followed as inpatient at our hospital, representing especially the sixth one. We look at the monthly distribution of cases, and we see that the number of cases began in April and peaked in July. You can see 125 cases also. And dropping to a quarter in August and disappearing from September onward. More than half of the cases are detected only in July. Located in the heart of the Cumhuriyet region, where the highest number of cases are observed, and our hospital also received a referral from other provinces like Yozgat, Tokat, Sivas, similar near cities also. And you can see Yozgat, Tokat, Giresun, Erzincan also. Yes, demographic, all things is okay, Dr. Robert? Hello?
Yeah.
Thank you very much. Can you see slides also?
Which slide?
Yani demographic characteristics also.
The characteristic,
And
maybe the case definition criteria.
Yeah, maybe yes. It is not prepared also. I don't know why. And demographic characteristic also age, median 56, and also these cases 38 female and 62 male person. And occupation is very interesting because 80% of them farming and other animal husbandry and health care workers, one person and one veterinary in these cases. Risk factors and 93% living in Crimean-Congo Hemorrhagic Fever endemic rural areas, also 93%, and history of travel to CCHF endemic rural area in weeks, 6%, and history of tick bite 66, and close contacts to animal 60, and close contacts to blood organ of an animal 34. Yes. And admission symptoms and signs for CCHF cases, you can see muscle headache 95%, 97% fatigue, and 60% splenomegalia, and 72% history of fevers.
Just a thing. Maybe we should, if maybe the last minutes, because maybe for time reasons, no, we are closing soon at some point the webinar here.
Yes, maybe I can finish. And these two cases, also patients were treated with intravenous fluids, blood products, and oral ribavirin, and maybe 50% of 25 cases died in first 7 days. And 30% people died maybe between 8 and 11 days also. Yes, and supportive treatments of Crimean-Congo hemorrhagic fever, IV fluid, electrolyte, also blood products, fresh protein plasma, thrombocyte suspension, erythrocyte suspension, mechanical ventilation, IV vasopressors, hemodialysis, and other H2 glucose and paracetamols. Yeah, maybe we can see that Crimean-Congo hemorrhagic fever cases are uncommon in Europe. They persist in endemic regions, Balkans, Russia, Turkey, and like Iraq. But sporadic local cases have been increasing in Western Europe, like Spain, Portugal, and Greece in recent years. Human cases are low across Europe, but the risk continues to increase due to the presence of tick and climate change. It is also beneficial to know that the number of cases in Europe which is not very high at the moment, is vulnerable to diseases that may be brought in by travelers from neighboring countries and regions where Crimean-Congo hemorrhagic fever is frequently seen, just as it is in Turkey, Iraq, Iran, and Balkan countries. Thank you very much.